Over the past few decades, many biological assays have been developed. Among them, next-generation sequencing, NGS, has made a big impact on biology. NGS refers to high-throughput technologies that allow millions of DNA or RNA fragments to be sequenced in parallel. In other words, instead of reading genetic information one piece at a time, NGS reads an enormous amount of information at once. This is like switching from reading one page at a time to scanning a whole book in minutes. Therefore, NGS enables genome-wide analysis at much higher speed and lower costs. RNA sequence allows us to identify cell types and cell states, track developmental processes, and study disease-associated transcriptional changes. However, here is a key limitation. RNA sequence mainly tells us where genes are expressed, not how their expression is regulated. So, if we want to understand the regulatory mechanisms controlling gene expression, such as enhancers, transcriptional factors, and chromatin structure, we need additional NGS-based approaches that profile chromatin and protein DNA interactions. This leads to the next methods: ATAC sequence and ChIP sequence. First, let's look at ATAC sequence. ATAC sequence is a method used to profile chromatin accessibility across the genome. It uses a hyperactive TN5 transposase, which is insert sequence adapters into open or nucleosome depleted chromatin regions. After transposition, the DNA fragments are amplified and sequenced, and regions with high read density are identified as accessible chromatin. These regions often correspond to promoters, enhancers, and other regulatory elements. To summarize, ATAC sequence provides a genome-wide map of where regulatory activity can potentially occur. This is like identifying open doors in the genome. But there is an important limitation. ATAC sequence alone does not tell us whether these open regions are actually active and functional. So to address this limitation, we need a next approach. Now, let's move on to ChIP sequence. ChIP sequence helps us determine which accessible regions are epigenetically active. In this method, chromatin is first cross-linked to preserve protein-DNA interactions, then fragmented and immunoprecipitated using antibodies against the specific protein. For example, ChIP sequence for histone modifications such as H3K27AC, H3K4ME3 reveals where these modifications occur in the genome, which helps us functionally annotate regulatory elements. Beyond histone modifications, ChIP sequence can also target regulatory proteins. One example is BRD4, a bromodomain-containing protein that binds acetylate histones and functions as a scaffold to recruit transcriptional factors and RNA polymerase II. ChIP sequence targeting BRD4 identifies regulatory regions that are not only accessible and accentuated, but are also actively engaged in transcription. BRD4 is frequently enriched at active enhancers and super enhancers, which drive high levels of gene expression. Thus, BRD4 ChIP sequence provides insights into which enhancers are functionally connected to transcriptional machinery, not just open, but actually active. In conclusion, NGS provides powerful tools to study gene regulation at multiple levels, while RNA sequence defines cell states and transcriptional outputs. Chromatin-based methods such as ATAC-seq and ChIP-seq reveal the regulatory architecture that controls gene expression. By combining these approaches, we can identify enhancer regions and understand how chromatin accessibility, epigenetic modifications, and transcriptional machinery work together to drive biological processes. Here are references. Thank you for listening.